of the lecture part. I'm not going to go through all of this tonight, go through maybe about half of it, and I'll turn you guys loose. You do have available to you, I have some thoughts on hard side if you want to look at them again, right, in nice cold water. I know you can work in your bottles and such with that. So let's look at the structure of what you've been studying and memorizing, and that's going to be blood vessels. And we can see by the diagram there's three major layers or tunics to your blood vessels. You have the tunica intima, right, because it is intimate with the blood. The blood will be right next to it. And we can look at what that tunica intima is actually comprised of. And it's comprised of, endothelium. And if you remember from 241, endothelium is the word we use to describe simple squamous epithelium that lines inside the blood vessels or the heart, kind of reconnecting to an earlier bit of information. But even if you forgot that, you should be able to look at this and go, oh yeah, that's simple squamous, I got that, no big deal. And then we have the tunica media, because it's the middle of our three, and the tunica externa, because it's on the outside. But now I want you, so nobody say anything, Kaz, hush, okay? I want you to compare the tunica media between an artery and a vein and tell me if they're different, and if they are, how, or are they the same? Compare the tunica media of an artery to the tunica media of a vein, and how are they different, if they are at all. It's a little bit of learning how to be critical in what you're examining, to look, not just look.
Yeah, I see.
How are they different? If they are, maybe they're the same.
They're
Kaz?
different, the elastic fiber.
So you see little black squiggly lines?
Yeah.
Right? Any black squiggly lines over here? Nope. What else is different, Scott?
Is it transitional on the artery and not?
Transitional?
Yeah, the transitional squamous.
Now how about the thickness? There's more here than there's over there.
Oh yeah.
So when you look at two blood vessels side by side, right? If these were, let's say the axillary vein and axillary artery, you can tell which is which because veins are always larger in lumen than the corresponding artery, and the walls of the artery are much thicker than the corresponding vein. Makes it much easier to identify which one is which in a cross section. The outer layer is the tunica externa, which is made up of connective tissue. You can see some stripes of elastin proteins in there, which make up the elastic fibers. And if you notice, one of the weird things about the vein is that they have these sporadic little smooth muscle cells that are placed in the tunica externa that are not found inside the tunica externa of an artery. So the presence of the elastic fibers in the media and the corresponding absence of those in the vein, the much larger lumen in a vein compared to the lumen of an artery, the much thicker arterial wall of an artery, the tunica media of an artery, and the much thinner tunica media of a vein are some of the key major differences. Now when we look at the flow of blood through all of our blood vessels, you are not going to work in here. You don't want to work in here for some reason, right? You don't want to work in here. Is this idea. Remember what I'm trying to have you guys think about in terms of why anything goes anywhere. It's the difference in gradients, right? Whether it's a concentration gradient or a pressure gradient. And we have the pressure gradient for why blood goes from one part of the body to another. And we talked about the idea of afterload on Monday night, and we talked about the peak pressure found in an artery was being 120 millimeters of mercury, for example. To absorb that huge amount of force, I want elastic arteries really close to the heart. So your aorta and your brachiocephalic, your common carotid, the left one and your left subclavian, are going to be made of elastic arteries to absorb that pulse of blood as stroke volume is pumped out. It's like having Scott sit on the middle of the waterbed and I jump on the waterbed. The big wave pushes you up, right? You want to be able to not go flying off the bed. You want to recoil with that flow of blood. And so this is our point of highest pressure. The pressure gets less and less the further from the source we get. And we tend to notice as our arteries start to get smaller and smaller and smaller. Well, when you go from elastic to just muscular, we lose that elasticity, that elastic tissue in the elastic artery. So by the time you get down to like a brachial artery, right, you'll be more of a muscular artery. And there's no point of demarcation. There's not a point where all of a sudden they go from this to this. It's like watching an ice cream cone. We just get less and less of this until we finally realize, oh, you have none of that. Okay. And we keep getting smaller and smaller and smaller down to the level of the arteriole. But notice the big difference between arterioles and muscular arteries. Don't anybody say anything out loud, but compare arterioles to muscular arteries and see if you can see what's different about them. And I'm hoping you notice immediately that the arteriole has no tunica externa and a greatly reduced tunica media. So much so that all you really have is a single layer of that smooth muscle tissue wrapping around the arteriole. That makes it easy for you to make that tube dilate and constrict. I only have one layer of tissue to manipulate above me. I can't do that very well with a muscular artery because I'm going to have to create folds and kinks in the externa. It's hard to make this get smaller or to stretch it out. But this is manipulable. So we can do a lot of our vasodilation, a lot of our vasoconstriction at the level of the arteriole. Because really, it's simply just endothelium and a single layer of smooth muscle tissue. Everything else has been taken away. Then we're going to transition down to capillaries, which if you notice how capillaries are different than arterioles, now all we have is endothelium. There is no smooth muscle around the outside, just a little tube of simple squamous, which we'll talk about in a moment. Then we're going to come over to the veins, and I realize venules and their cousins, the arterioles. How are they different? Well, these guys have no smooth muscle tissue. They do have an externa. These guys don't have an externa, but they do have a thin layer of smooth muscle tissue. The lumens are pretty much the same. Right? It's more of a structural difference between these two guys. But then I start moving more and more towards the heart, getting closer and closer, further away from the capillary bed. Now I begin to have all three layers that are present, just as I do in a muscular artery, but my lumens are larger compared to its cousin of a muscular artery. We start getting even closer to the heart, further and further from the capillary bed. We have now a much larger lumen. And this will be the size, almost an inch in diameter. Your thumb is a good measurement for the superior vena cava and the aorta. And you maybe saw that when you did the dissections on Monday, where putting your finger in the superior vena cava, your whole finger fit in there quite nicely, up in the aorta, right? That gives you a good idea of how big those vessels are. If I want you guys to think about this being a high source of, or high place of pressure, like 120 millimeters of mercury, and I'm trying to press upon you, things go from high to low, 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 higher to lower, higher to lower. By the time we get to here and here, we're down to about 2 millimeters of mercury of pressure. So the whole thing is designed to be a downhill flow from high to low, high to low. The way you can really appreciate that is if you didn't really notice on Monday night how thick the aorta was, right, and how thin the superior vena cava was, you can actually see the color of your glove through the superior vena cava's endothelium tissues. So that's not very thick at all. It doesn't need to be thick, it's a low pressure system. So don't get fooled by, oh, it's a really thick vein. No, I mean, that was less than a fraction of a millimeter in thickness. That still has all three layers. All right, let's look at our three kinds of capillaries that we have. But first, let's give you the function of capillaries. That is a place of diffusion between the blood and the tissues. It's a place of diffusion between the blood and the tissues. You can give them nutrients like glucose. You can give them oxygen. And then we can pick up their carbon dioxide, we can pick up their metabolic wastes. And it's easy to do because we're looking at passing through a simple squamous cell. That's a very thin wall to be able to deal with. We're going to be looking at three types of capillaries. The first one is called continuous. And here's the way to think about it. If I was a red blood cell, my little red light, traveling through this capillary, I realize that the inside, or even the outside, is a nice, continuous, smooth surface. I don't see any holes in it. Right? And this is your most abundant type of capillary, but it's also the least permeable. It's the most abundant, but the least permeable of the three. But it's still pretty permeable, otherwise our cells wouldn't get oxygen and nutrients. It's just the worst of the three that we have. And they're found everywhere, and I really should remove this from the slide just to give you guys grief, except for cartilage epithelia. Remember, cartilage is avascular. That's why it doesn't grow and repair itself. And how do we define epithelia? Do you remember?
Some the
That
rough
lines,
on the
tissue
side.
that covers the outside and lines the inside of the structure.
with a basement membrane between it and the vascular tissue.
So is this epithelia right here?
Yes, yeah, yeah,
Inside of my stomach where my cheeseburger would be sitting?
yeah.
Lining my small intestine where my cheeseburger will be digested?
Yeah.
Right. And are there blood vessels there? No. Right? There is never blood supply in epithelia. Right below it, yes. Remember the dermis? Right? And the papillary portion of the dermis, that's where your capillary bed was. Right below the stratum basal. But there was no capillaries in the epidermis, which is epithelium. Now, compared to that, let's get to the little more porous capillaries. And that's going to be these guys called fenestrated because they have fenestrations. Looks like somebody took a shotgun and poked lots of holes in it. That increases the ability for things to diffuse in and out. So it's way more porous than a continuous capillary. Where would I have really porous capillaries? I really want to make sure that things can get into it and get out of it. How about the anteropituitary, where my hypophysis portal system is, so I can get those releasing hormones and release my stimulating hormones. Anteropituitary has has fenestrated capillaries. Your kidneys have fenestrated capillaries. Remember, those holes are still too small for red blood cells to pass through, unless you get them irritated due to long distance running. But still, those pores are really good for letting solids pass through, but they're still too small for a red blood cell to fit through. The third kind is known as sinusoids. Now, if you look at it, yeah, they're fenestrated, but they also have gaps between the actual cells. You can drive a Mack truck through these things. These are really porous. And as far as we know, they only exist in your liver. And we will revisit that when we get into your hepatocytes and digestive processes.
So you don't have them in your bone marrow where the new blood cells have to get in?
Nope. As far as we know, it's only in the liver. So that gives you three kinds of capillaries, right? Continuous, fenestrated, and sinusoidal. Continuous are the least porous of the three, but still pretty porous. Otherwise, we wouldn't get oxygen, carbon dioxide, and nutrients to our needing cells. Now, speaking of that, we need to look at a capillary bed. And also a little more practice in learning to learn how to look at things. So I look at the diagram and I realize, okay, so there's an arteriole, which I remember is nothing but endothelium wrapped with a single layer of smooth muscle cells. This is a cross-section of that arteriole right there. But nobody's saying anything. But I want you to think about how would you describe what they're trying to show you? What did they do for you to see this particular view that you're looking at? Don't say it out loud, but what did they do to give you that view? What is the view they're showing you? What they did is they took your blood vessel and they cut it right in half along the entire line, like a big long blade, and they removed the top half. You're looking down into those blood vessels. So let me ask you this. What in the heck are these little pink things right here? Don't say it out loud. What are these stupid little pink dots? What are those damn things? Scott, do you know? What are those things?
You know, right now.
Dave, do you know?
Maybe somatic cells that do
Nope.
stuff? No?
So again, how did we make this view? What did I say we did? Took a big knife and cut all this in half, right? Like cutting this in half
It's
Oh,
and
those are the little
removing the
muscles.
top half.
smooth muscle.
That right
Smooth
there
muscle
is
cells.
the looking down on the end of the smooth muscle. These are all just the ends of the smooth muscles that you cut in half. Which means the smooth muscles are wrapping all the way around here, and all the way around there, and all the way around this guy right here. But look where they're not. They're not over here. On the venule side, on the venous side of the capillary bed, there are no smooth muscle cells. only on the arterial side. That's why I'm trying to teach you guys how to look. And like, what the hell? Don't just look, figure it out. So it makes a little more contextual sense for you guys. So why is that important to realize that these are all smooth muscle cells that we're looking at? Well, at the entrance of every possible pathway from the arteriole, like you're leaving the highway and taking the exit ramp, right? Each ramp is lined with precapillary sphincters. Sphincters, that means they're comprised of circular bands of smooth muscle tissue. They wrap all the way around to create a sphincter. Which means I can constrict it and close it off, or I can relax it and let stuff flow through. Do you guys remember the different patterns of orientation of skeletal muscle cells? Patterns of orientation of skeletal muscle cells? You got pinnate, convergent, right? Parallel, circular, right? And this is an example of circular orientation of muscle cells in this context. Now, that's important because we need the ability to kind of do traffic control. I may need to let blood flow through here, but not let it flow through there or here. So I can constrict that pathway, relax or leave alone that one, and constrict this one. And the blood that tries to go there, the street's closed, I can't go there, I'm going to go down through here. Or I can constrict this one, open up the two end ones, whatever combination you want. Because, let me pull this screen up so I can put a diagram on the whiteboard. So there's our capillary bed, and there's a branch of the capillary bed here. This is tissue. And in there I'm going to have 3 cells. I'm going to put one cell here, I'm going to put one cell there, I'm going to put one cell right here. Give them little nucleuses. And the little buggers are hungry. They need oxygen. So what if I look at an oxygen molecule traveling down this pathway, and the O2 is separated from that cell by just simple squamous epithelium? Well, that's pretty close. I can diffuse to that no problem. So I can feed you. And if I diffuse it to here, I can probably supply you with oxygen as well. Now, I may or may not be able to reach that guy. And that guy's going, well, what about me? That's not fair. I'm not getting my share of oxygen or glucose if you want. That's not fair. Well, don't worry. Aren't you right next to this avenue instead of this avenue? So the O2 that's in here, right, could diffuse and feed that cell, or it could go in and feed that one. See what I'm saying with that sharingness? Now, the problem is, you have way more cells than we do blood to supply them. So it's like having 2 tables and 50 guests. You gotta take turns. So we redirect the flow of blood. That's called vasomotion. I will alternately change the flow of blood through the capillary bed. I will maybe constrict this one and relax that one so it flows here and here. Then after about a second or two, I'm going to close off this one and open up this one perhaps. You might do this 30 or 40 times in a minute for any one capillary bed. Simply because we don't have enough blood to feed everybody, so I just simply take turns. You get your share, then we let you get your share, then you guys get your share, then, on and on and on. Now, if you need to really not send blood to the cells at all, maybe due to temperature issues or for some reason, your body says, I'm going to sacrifice these little buggers here, because I need to send blood back pronto to John's heart or his lung or his brain or something, I can constrict, and dilate this arterial venous, artery to vein, anastomosis. It's a shortcut, or if you want, it's the business loop. Right? I go around all the cities and businesses. It's a direct connection from arterial to vein. There's no feeding because look what's wrapped around you. Smooth muscle tissue. You can't do diffusion through these little joints. So this is not designed to feed cells. It's designed to get blood quickly from the arterial side to the venous without screwing around. now comes perhaps the hardest part. It's not really hard, but it does seem to be the stumbling block for this. And to help you guys have room to write all this stuff down, you have this image in your notes, then you have this one. So you have room to write notes for this part, and you have room to write notes for this part, right? But I'll be looking at the whole thing on one big screen. All right. Once I get you guys to understand this, then respiration, digestion, and the renal system is a piece of cake, because they all do the same thing. This is the foundational idea, sort of like releasing hormones and stimulating hormones. You know about one, you can figure out the others. There comes the blood down an arteriole, which you now know has just a single wrapping of smooth muscle tissue, right, into a capillary, which has no smooth muscle wrapping around it. And there's blood pressure on the arterial side of this capillary. And the blood pressure is 35 millimeters of mercury. And that's known as CHP. Now, CHP does not stand for California Highway Patrol. Anybody from California, right? They're not chips. But it does stand for capillary. And the one word I do not like, right? CHP, capillary hydrostatic pressure. What does hydro refer to?
Water.
What does static refer to?
Doesn't
Not
change.
movement.
Yeah. Does your blood pressure not change? No, right? And I was raised by a fluid dynamics specialist at Stanford, which really spoiled me. This really should be called capillary hydrodynamic pressure, to be quite honest, because it's constantly in change, right? It's dynamic, it's never the same. However, be it as it may, this is the tools you have to work with. Capillary hydrostatic pressure is the blood pressure in the capillary. And what is blood mainly comprised of? Water. So it's just the water pressure against the walls of your capillary. It's how hard they're pushing against the wall. Think about just for a moment an everyday word you guys are very familiar with, maybe. Water pressure. Right? What is water pressure? It's the pressure water is pushing against the pipes, right? It's the same thing here. It's our biological water pressure. It's just the pressure of the blood pushing against the walls of our blood vessels. So if this is your blood vessel, right, and then there's fluid pushing against the wall, we now know how hard it's pushing. We measure it as 35 millimeters of force. Now, in the blood is this other term over here that they somewhat defined. They left an important word out. It really isn't BOP. It should be actually BCOP, which stands for blood colloidal osmotic pressure.
So would you write like as no, not BOP, like the
Well,
COP?
think about this. They wrote BCOP here, right? They just forgot to put the C in the legend. Now, colloids are albumins. Those are the proteins I named in tonight's lecture about composition of proteins in blood. They're just the albumins. They're proteins that are way too big to leave. They will always be in the capillary. Now, why is that significant? Look at the last part, or the second to last part. I have an arrow next to you. When you hear the word osmotic, what comes to mind?
Water.
Water. What else comes to mind when you hear the word osmotic or osmosis?
Solution.
Solids. What other word comes to mind? You're on the right track.
Solution.
Solution.
Permeable membrane?
Yeah, that too. That's good.
Movement of water.
Movement of water. Which way will water go?
Oh, ions.
On the high still.
But how do you know where there's high amounts of water? What vocabulary words would you use?
Concentration.
No?
Hyper.
Hypertonic, hypotonic, isotonic. As soon as you hear the word osmotic, you're thinking about semipermeable membranes and the movement of water and hyper and hypotonic solutions. They should all just be pouring into your brain. So I will use that vocabulary to figure out where water is going to go. Now, what does hypertonic refer to? What does it mean?
Greater concentration of solutes.
compared to solvent, right? So there's more dissolved particles than there is solution.
Right, and compared to the other side of the
Compared to the other side. So if one side is hyper, the other is always hypo, right? So I have a certain degree of osmolarity or of solutes in my blood. Due to the albumins, the proteins, they're trying to attract water towards them. And they're attracting with 25 units of force. I just keep it simple, it's like physics. I have one force pushing out at 35 units of force. I've got one force trying to keep the fluid from leaving, and it's using 25 units of force. Which force is greater? The the CHP is, right? The blood pressure is greater. So the net difference is 10 millimeters of mercury of pushing fluid out of the capillaries. Well, how much fluid in a day actually leaves through capillaries?
It says 3.6 liters on the chart.
24.
24.
Oh, 24.
Does that sound right to you?
No.
Sure, it comes
How many
back.
liters of blood do you have?
5.
About 5 or 6 maybe, right?
Well,
But this is correct. It's just going around and around and around and around all day long. Plus, there's something else to think about.
New blood.
Filtration is the word we describe to filter the fluid, right, through the wall like you would filter coffee, right? And so the solution comes out carrying its nutrients with it and oxygen into the neighboring interstitial space. And how much fluid gets in there? Well, apparently 24 liters a day. But at the same time, as you have that fluid leaving the capillaries, your volume diminishes. That's in here. And with less volume, we get less pressure. Look what didn't change. The B-cop didn't change. The proteins can't leave. So their attraction is always the same all the way across. So by the time the blood is moving through here, we're filtering out plasma, mainly the water part. The volume of blood gets less and less compared to this side and this side. The pressure drops, and now who's the greater force? The B-cop is, right? Now there's a greater osmotic pull, actually pulling fluid back into the capillary bed. Most of this fluid that leaked out got pulled right back in. Now we call that reabsorption, and you have to be specific. Don't call it absorption. All right, this is term specific. Because when you read something, what are you doing? Like if I recycle it,
Taking it back.
we're taking it back, right? So here's what I want you to think about this. I eat a cheeseburger, it goes in my stomach, goes in my intestines, then I absorb the nutrient. It's the first time it's coming into my blood, so it's absorbed. Right? It goes in my blood, it's in my blood, little cheeseburger particles. Now the cheeseburger particles got filtered out, and I got cheeseburger particles in my tissues. I then pulled the cheeseburger particles back into my blood. It's the second time it got in there. It was reabsorbed. Right, so it's a very term-specific thing. Not absorb, but reabsorb. And how much fluid do we reabsorb in a day?
20.4 liters.
But we filter 24. That doesn't seem okay. So if I keep filtering out more than I reabsorb, I'm going to become like the giant Stay Puft marshmallow, right? It's big and puffy. How come I'm not big and puffy and swollen? Because the excess difference, that 3.6 liters, actually goes into another set of vessels called lymphatics. So there's actually no net gain or loss. 24 in, 20.4 in, and 3.6 in, that matches the 24. And as long as that excess fluid that was not reabsorbed can enter the lymphatics, is where does this fluid go? What does it say right there? Into circulation, which we'll probably describe next lecture, but it's actually your subclavian veins. It gets reintroduced, so there's actually no net loss in volume. Now, it's possible for it to accumulate in here, what we call edema, right? And for the first time in my life, I actually got to experience this. So I suffered greatly at all the expense I had for 241 this summer. I had to get your grades out really fast. I get my ass to Idaho. I spent 12 hours in my office chair at home grading your exams, calculating grades, and submitting them. I got up twice for coffee and pee, and that was it, right? And the second time I went, holy shit, oh my God, I'm getting old, holy crap. Where'd my toes go? Where'd my veins go? I could poke my foot and get pitting edema. My ankles were swollen. My foot was all puffed. Went, holy shit, I'm getting old. Oh my God. So what did I do? 2 ways to reduce the edema. I walked around for about an hour. Went to the park and walked 3 miles. Right? That walking, the musculoskeletal pump, helped to push the fluid into my lymphatics, and that got pushed into my veins, and that fluid got drained out. What I really should have done is called Domino's, have them deliver food and have somebody deliver beer for me, lay on the couch, prop my feet up so they're above my heart, and let gravity do all the work as I eat pizza. It would do the same thing, because gravity would have pulled the fluid into my veins. So many of you work in geriatrics. You get people with edema, right? One of the things you do for them, you get them to walk around, become physically active, maybe give them a massage, right? To help push the fluid into the lymphatics.
And the diuretics, which beer is, a diuretic.
That's, yeah, which we'll get into. That's why I wanted the beer, okay?
And the salt and the protein in the pizza also help you maintain your human levels and increase keep the tendency, the BCOP up.
The BPOP? Yeah, okay.
BCOP. Well, it's a new word for me. Not
Yeah,
for you.
okay. So does this process make sense? Right, do you understand why we get filtration? Because the greater blood pressure compared to the osmotic pull of the proteins in your blood, right? That's a greater force than this, you get a net loss. But now that force diminishes due to the loss of blood volume, which makes the pressure drop. This pressure does not drop because the proteins, they can't leave, they're always in there. They always have an osmotic pull trying to keep the fluid in. But now this force is greater than that force, so fluid is actually coming back into my capillary bed. This whole concept is going to do what happens in respiration, digestion, and the renal system. So I want you guys to get really comfortable with it and understand why things are happening. Because you're now getting to the point where it isn't enough just to memorize, but to understand why things do what they do. And then you can problem solve, you can figure things out. So work on that part. If we need to help you with that, we'll be happy to do so. Almost done for tonight. Since veins are a very low pressure system, I need to get the blood back to my heart. And veins have one other thing that arteries don't have. They have valves in them, like stopgaps, like points of no return or no backing up. They look just like your semilunar valves. Picture the semilunar valves you guys found on Monday night. And that's what these valves look like. They are an extension of the tunica intima. They're a part of that tissue. And they're designed so that when blood actually passes by them, right, they open up, the fluid moves through, and then as soon as blood tries to fall back down again, like the semilunars, they catch the edges of the valve, they open, or they billow out, and they seal shut. So this volume of blood can't go to the previous section of the vein. But if the valve fails, then as the blood that came from a lower section enters the new section, and the section above it fails, I get now twice the volume in this section. And maybe even more than that if multiple vein valve fails. I get to have the blood pull and pool and pool. And you start getting bulging of the veins because you've now doubled or tripled the volume in that one little segment. So it can look like that. All right, that's one big, nice, puffy vein. So one way is you can wear compression socks, right, to help keep the pressure so the fluid can't accumulate in there. You can get them walking around, right? But the problem is that it's the chronic, long-term problem. It isn't just an overnight issue. But after years of having fluid accumulate in the veins and stressing the valves will fail, the fluid will build up. And a lot of people don't like having varicose veins. It's not very attractive. So what surgeons can do is they can take a laser or a scalpel, they can maybe cut this part here, and they can then cut that part right there and take the forceps and like a big long spaghetti noodle and just pull the whole thing right out. All right? Because you don't need it. The one cool thing about your vascular structure for veins, you have way more veins than you do arteries. So if I lose that one path, no big deal, I'll take another path. I will still get to the femoral vein, the great saphenous, whatever vein I want to get to. I'll just take an alternate route. Or they can cauterize it. Right? But they still will give you lots of alternate routes. So it's not a big issue to have one or two of them taken out.
So is it kind of like the same thing where you're seeing like the purple varicose veins?
Yep, exactly.
Bulge? It could just be
Yep.
through the seed of the vein? essentially?
Yeah.
Okay.
Yeah. And something is due to
Is there a reason why it doesn't bulge? If it's just shown more prone?
more muscular walls,
Okay.
right? They're probably more like surface arteries, which is why they're more purplish red than the veins are. Last little bit that we're gonna quit for the night. We can use your blood pressure, or we can actually control your blood pressure from one aspect. There's multiple aspects. We're only addressing one. And that's gonna be through the use of the sympathetic nervous system. And if I want to use the sympathetic nervous system to vasoconstrict, right, I'm gonna make the arterial smooth muscles constrict. They will constrict. They will contract, which means the diameter gets smaller. And they'll be using norepinephrine to do this. The neurotransmitter, not the hormone. So a nerve goes to all of your blood vessels. Well, many nerves do. If you think about your blood pressure, 120/80. We talked about that in terms of the idea of afterload for cardiac output, but I also asked you guys, well, why is it never 0? If it's in between heartbeats, what's helping to maintain that pressure? And it's vasoconstriction. You have what's called vagal tone. There's a certain level of stimulus to all of your blood vessels to keep a certain amount of constriction going, to maintain a certain amount of pressure. And some folks who have high blood pressure have overactive sympathetic nervous systems. They're just sending too many signals, which makes the blood vessel constrict more than it's supposed to, which raises your blood pressure. So one way is to give them what's called an alpha blocker or a beta blocker. Just keep the norepinephrine from landing on the receptors. If norepinephrine can't land on the smooth muscles, then they can't constrict. They end up dilating, which is the relaxation of this. So by sending fewer signals, they get to relax the blood vessel. That's what's called vasoconstriction. We're still using the same symptom. This isn't like sympathetic versus parasympathetic. This is just sympathetic signals and then the lack of signals for the dilation part. So increase the signals for constriction, decrease the signals for dilation. That's one way to do this. That's the only way you have to worry about. And that's where we stop. We'll get to the, whoops, wrong direction. We'll get the rest of this stuff next time. Why are you doing it this way? Stop that, there we go.
Is that first
All right.
gonna be on the test on Monday?
Yeah.
Do what, huh?
Is that first half gonna be on the test on Monday?
Yeah, it's in the study guide.
Yeah.
I've got
Well, what
that
about the
far.
second half? No, obviously, duh. Okay, cool.
All right, so you guys have your blood vessels and models to work